Bismillahirrahmanirrahim. Alhamdulillah, sallallahu alaihi wasallam. First of all, we would like you to come for the seminar today. This is the result of the research of the PhD student in plant protection, and his name is Muhammad Taimoor. Just I want to give a brief about Muhammad Taimoor. He born in 23rd of January 1986. While I graduated from this college. He's married with no kids yet. And he got his BS in plant pathology in 2007, the GPA 3.3 out of 4. He got his master in plant pathology in 2009 from Arid Agriculture University, Pakistan, with a GPA 3.7 out of 4. And he joined our department in King Saud University for the PhD in our lab in February 2012. And his GPA 4.8 out of 5. And he did around 8 publications. And he has 5 research articles under review now. The title of his research is Molecular Characterization of Cranial Viruses Involved in the Etiology of the Yellowing Disease of Tomato and Cucurbit Crops and Role of the Arable Weeds in their Epidemiology in Riyadh region of Saudi Arabia. Welcome, Taimoor, good luck. 20 to 25 minutes.
Thank you very much, Doctor, for my introduction, and thank you all for coming. Good morning. Bismillahirrahmanirrahim. Doctor has already described the title, so we will go directly to the introduction. So the most important crops in Saudi Arabia and worldwide, we will go for the introduction. We consider 2 crops, tomato and cucumber. These both crops are very important in worldwide and in Saudi Arabia because of their daily usage. And in Saudi Arabia, 500,000 tons of tomato are produced from around 16,000 hectares, and around 1 million tons of cucumber are produced from 39,000 hectares. And these both crops are suffering from yellowing disease. This yellowing disease is explained as a general yellowing of the crop in the greenhouses. This yellowing may be because of different reasons, but the most important reasons are the viral reasons, and we will be looking for the cranial viruses as an agent of this yellowing disease. So, the cranial viruses are flacious rod viruses, and they belong to family Cholesteriviridae. These are phloem-limited viruses, and they have a segmented genome. The RNA is single-stranded positive sense, and the RNA1 is replication. It is involved in the replication of in the proteins that are important for replication, and RNA2 is involved in the coding of different proteins which are involved in cell-to-cell movement and vector transmission. So the members of this genus which we are looking are tomato chlorosis virus, tomato infectious chlorosis virus, cucurbit yellow stunting disorder virus, and cucurbit chlorotic yellow virus. Tomato chlorosis virus and TOCV and DICV are infecting tomatoes, and CYCV and CCYV are infecting cucumbers. The host range of tomato infecting cranial viruses are wide, and still this is expanding. They are, in fact, the new hosts are being identified still to be infected by these viruses. Already identified hosts are belonging to 7 different plant families, and several species of weeds are already identified as a host of this virus, and still we are getting more, as we already identified few more hosts for belonging to weed species. And the differential host for the identification of tomato chlorosis virus is lettuce, as in greenhouse if we have to identify or differentiate between tomato chlorosis virus and tomato infectious chlorosis virus, we use lettuce as a differential host, because tomato chlorosis virus is not infecting lettuce and TICV does. On the other hand, the cucurbit cranial viruses, CCYV is infecting again different families. Around 6 families are infected by CCYV and 8 families are infected by CYSVV. The most important character of these viruses are the transmission. These viruses are transmitted in a semi-persistent manner. As these viruses are phloem-emitted, they are transmitted by 4 different species of whiteflies. Trialrodus reporiorum, Bemisia argentifoli, Bemisia tabasi and its biotypes, and Trialrodus avitilonia. The most important one is the Bemisia tabasi. That is important because you can see it is transmitting TOCV, CCY and CYSVV, all of them. On the other hand, TICV is only transmitted by Trialrodus reporiorum. The unique character of TOCV is it is being transmitted by 4 species, and this makes this virus more established and well transmitted in different regions of the world. None of these viruses is reported to be transmitted by seeds or mechanical means. On the basis of field observations, we started our study in 2014. What we observed in the greenhouses, we found that there is a yellowing disease, the general symptoms of yellowing, and also we found high population of whiteflies on the undersides of the leaves, and also we found a high number of weeds that are prevailing inside the greenhouses. And also we observed the symptoms that are very similar to those caused by the greeny viruses. So the symptoms on cucumber, we observed that there is intraveinal chlorosis that starts from yellowing spots in between the veins, and at the later stages, these yellowing spots develop and merge together and become very clear yellowing symptoms, and the veins remain green. And in the severe conditions, the necrotic specks also develop. These symptoms are very similar to nutritional deficiency, but these symptoms never reach the new growth. They are always confined to the older leaves. The leaves become brittle and slightly curled. Similar symptoms were observed in tomato here. There are intraveinal chlorosis symptoms and the necrotic flex and brittleness of the older leaves. Then we had some observations in the vicinity of the greenhouses, and we found Malva parviflora and some other weeds also growing there. This is very clear that this is the most dangerous situation here, that this high population of weeds is growing at the openings of the greenhouses, and you can see all of the greenhouses here are having this weed in the openings. Again, Lectuca saligna, Lectuca cereula and other grasses are growing in the sides of the greenhouses. This is another picture of Malva parviflora that is growing inside in the vicinity of the greenhouses. These all weeds are proved to be the source of the virus. So this is the most important situation to look at, that these are the source of the virus when the crop is not there in the greenhouses, and when we start the growing season, they work as a source. On the basis of these observations, we designed some objectives. So the objectives included the survey of tomato and cucurbit plants exhibiting yellowing symptoms and identify the causal agents in Riyadh region. Study the host range of greeny viruses involved in the etiology of yellowing disease in tomato and cucurbit crops, and it includes both cultivated crop plants and arable weeds. Determination of phylogenetic relationship between the Saudi isolates of identified viruses and other isolates reported worldwide. Identification of whitefly species and biotypes associated with the yellowing disease, and evaluation of the transmission efficiency of greeny viruses using the identified biotypes. To fulfill these objectives, we designed some material methods. First of all, we did collection of samples, and this included 228 samples from tomato, 43 samples from pepper, 33 samples from beans, and 9 samples from eggplant. These all samples were tested for the presence of TOCV and TICV. To detect CYSVV and CCYV, we collected 134 cucumber samples. And apart from this, we collected 324 wheat samples, and these were tested for these 3-4 viruses. This was an interesting situation. We found a bitter gourd plant that is a volunteer plant, and this plant gave us some unexpected results. As we collected them because we found some unique symptoms. These symptoms were yellow spots, and this was systemic in all the plant. Apart from plant samples, we collected 34 whitefly samples using handheld aspirator, and we preserved these samples in 96% of ethanol. After collection of samples, we detected the presence of these viruses using molecular techniques. ELISA is not a good option to detect the cleaning viruses because the antibodies has to be produced, and as the virus titer is very low in the phloem, so the quality of antibodies produced against them is not good for the testing. So the only method for identification of these viruses is molecular techniques. For this, we did RT-PCR after RNA extraction. For detection of tomato-infecting cleaning viruses, we considered the heat shock protein region, and first we identified that with the general primer. The expected size for this primer was 587 base pairs, and we used this amplicon as a template for nested primer. And in nested primer, we detected TICV and TOCV using the specific primers. And the amplification size of TICV was 223 base pair, and TOCV was 463 base pairs. For the detection of cucurbit-infecting cleaning viruses, the RNA was extracted using the same method and was identified by specific primers. For COSDV, we again considered heat shock region, and the amplified product was 364 base pairs, while for CCYV, we had the primer that was targeting cold protein region, and the amplification size was 760 base pairs. To identify the whiteflies, we extracted DNA from single insect separately. For the extraction of DNA, first of all, the ethanol in which they was preserved was dried at 45 degrees centigrade, and after that they were boiled at 95 degrees centigrade in 25 microliter of 50 molar sodium hydroxide for 20 minutes, and after that they were neutralized. The solution was neutralized with the same amount of Chris HCl, and after centrifugation, 3 microliter of the supernatant was used as template for downstream PCR reactions. For identification of Babesia tabasi, we targeted mitochondrial gene, and the specific primer was used, and the amplification size was 800 base pairs. After identification of these viruses and insects, we did phylogenetic analysis using DNA star program, and 40 isolates from TOCV and 13 isolates from CCYV, 5 isolates from CYSDV, and 3 whitefly samples were selected to identify as on the sequences base, and the sequences were done from Advanced Genetic Technologies Center USA and PG China, and these all isolates are selected on the basis of host and location they were collected from. After completion of the lab work, we started the host range experiment for CCYV in the greenhouses. So to carry out the host range experiment, we had to establish a controlled environment. To establish the controlled environment, we had insect-free cages, and all the plants were maintained in them before and after inoculation. The plants from all the seedlings from the representative species included in the greenhouse were grown in the controlled environment to avoid any unwanted transmission of viruses by insects. This is the virus-free whitefly colony that was collected from field and was transferred to tomato plant that was non-host to CCYV, and we kept it for 2 months to ensure the virus-free status of these whiteflies, and then we established also an infected CCYV plant. And we use this plant as a source for downstream transmission experiments and also the host range experiments. To make these whiteflies viruliferous, we allowed them to feed on this positive plant for 48 hours of acquisition access period. And to carry out the host range experiments, 40 insects that were supposed to be viruliferous were, after the acquisition from infected plant, they were confined to each treatment of the healthy plants of belonging to these all 16 species of plants. And they were allowed to inoculate these plants for 72 hours, and each treatment was replicated 10 times, and control inoculation was done using virus-free whiteflies. transmission efficiency of the identified whitefly biotype, we did first the determination of minimum number of whiteflies that is enough to transfer the virus from infected plant to the healthy plant. For this purpose, the virus-carrying whiteflies were allowed to feed the healthy plants. In first treatment, we allowed one whitefly to inoculate one plant, then five whiteflies for one plant, 10, 20, 40, and 60 whiteflies were confined to each plant to identify which is the minimum number of whiteflies that is enough to transfer the virus. To determine the acquisition access period, virus-free whiteflies were allowed to feed on the CCYV infected plant for 30 minutes, 60 minutes, 90 minutes, up to 48 hours, and after completion of the acquisition access period, these whiteflies were transferred to healthy plants of cucumber and were allowed to inoculate them for a fixed period of 72 hours. Next, we identified the inoculation access period. For inoculation access period, we fixed the acquisition access period for 72 hours, and then we pulled out these whiteflies and transferred them to each set of the cucumber plants and allowed to inoculate them for 30 minutes, 60, and up to 48 hours. To determine the persistence of the virus in the vector, we allowed the vector to feed on CCYV positive plant for 72 hours, and then we transferred these plants to the healthy plants for 24 hours. After 24 hours, we take these whiteflies and transfer them to a healthy next set of the plants, and then we did serial transmissions up till 10th day. So this is to check how many days after acquisition the whitefly is able to transmit the virus to the healthy plants. To determine the host efficiency, we selected two hosts, the Tura and cucumber. These are both infected plants with CCYV. We allowed the virus-free whiteflies to acquire virus from these both plants for 48 hours, and then we transferred these whiteflies to 10 healthy plants for each, the Tura and cucumber. Again here, source is cucumber, and then we transfer to the Tura and cucumber. Coming to the results, we found that we tested 784 samples in total, including all the hosts. For tomato, we tested 228 samples and we found 202 out of them were positive, and all of them were positive for TOCV, 26 were negative. And cultivated crops, these were tested for the presence of TOCV, so we found that out of 98 samples, we found 39 samples are positive, and these 39 samples include bitter gourd, pepper, beans, eggplant, and 59 of them are negative. Regarding the cucumber samples, out of 134, 120 were positive and 14 were negative, while this 120 included 82 samples infected with CCYV, 26 samples with CYSDV, and 12 of them had mixed infection with both CYSDV and CCYV. Out of 324 weeds, we found 103 were positive and 221 were negative. Out of 103 samples, 87 were infected with TOCV, and 13 were infected with CCYV, and 3 samples were infected with CYSDV. None of the samples were infected with DICV. The representative gels of gel electrophoresis showed that this is the results of TOCV from wheat, from cultivated host, and from tomato, and we found that the specific band of 463 base pairs was amplified. For CYSDV, we had a specific band amplified at a size of 364 base pairs, and for CCYV, we had a specific band for 760 base pairs. As a whole, we found some new hosts from weeds, and these all species which are mentioned in white are first time identified hosts of TOCV worldwide, and the red ones are the first time identified hosts from Saudi Arabia. For CCYV, these are the newly identified weed hosts, and for CYSDV, only one weed was found to be infected with this virus, that is the Marburg parviflora. Looking into the phylogenetics analysis of TOCV, this is the characteristic feature of coronaviruses, that they have low genetic diversity. As this is very clear from this, that all the isolates, we have 40 isolates of TOCV from Saudi Arabia that are compared to 20 isolates from worldwide, and all are making one bigger clade. And this is regardless of the host they are collected and regardless of the location from which they are collected. Again, these are the Saudi Arabian isolates, and this is very clear, they are isolated from different hosts and they are sharing the same clade. Interestingly, we have two isolates, one from Chenopodium morel and one from Malva pyriflora. They are divergent and they are expected to be another strain of this virus because they have 78% nucleotide similarity. So looking into them in details would be interesting. The phylogenetic analysis of CCYV showed the same trend, that there is low genetic diversity and there is no apparent role of host or location from which they were collected, and they are falling close to each other. CYSDV, we analyzed 5 Saudi Arabian isolates, and we found that the previously known phenomena for this virus, that this virus is having two subpopulations, the eastern subpopulation and the western subpopulation. The eastern subpopulation is confined only to Saudi Arabia, and this shows that this virus is endemic, and the genetic makeup is somehow different from the other isolates. For identification of whiteflies, on the basis of PCR, we found that the whiteflies we collected are all Bemisia levasi, and to identify the biotypes, we did the amino acid based analysis, and we found that the isolates from Saudi Arabia are 100% similar to the already reported biotype B. So we conclude from here that the whitefly prevailing here in Saudi Arabia is belonging to biotype B of Bemisia levasi. The host range results of CCYV showed different type of symptoms, like the Chenopodium coenova and Chenopodium amnioticulus show very mild symptoms, and these symptoms were observed a little late as compared to the cucurbit host. Cucurbit hosts, all of them show good symptoms, and here the middle board, this is clear yellowing as compared to the healthy one, and this is the dura showing intervenal chlorosis, yellowing on Nicotiana glutinosa. Cucumber is very clearly showing the typical symptoms. This is bottle gourd, melon, and pumpkin. Coming towards the results of transmission efficiency, we found that one whitefly is enough to acquire and transmit the virus to the healthy plants. And also, when we determined the acquisition access period and inoculation access period, we found that 30 minutes and 60 minutes is not enough, but 90 minutes is enough for the whitefly to acquire the virus from the infected plant, and again another 90 minutes would be enough for the virus to inoculate for the whitefly to inoculate the virus into the healthy plants. To determine the retention of the virus, we found that after 6 days of acquisition, the whitefly lost its ability to transfer the virus to the healthy plants. This is clear from here that up to 6 days we found the amplification of specific bands, and after that there is no amplification and there were no symptoms on the plants as well. When we identified the efficiency of host, we found that when we used cucumber as a source, this is mentioned in the orange line, we found 90% of the plants that got infected when we used cucumber as a source. When we used cucumber as a source and inoculated the tura, we found again 90%, but on the other hand, when we use the tura as a source, the infection level goes significantly low. Similarly, again when we use the tura and inoculated against the tura plants, the infection level was very low. Concluding this research, coronaviruses either infecting tomato or cucurbits are well established. TOCV was identified in 89% of the tested tomato samples, 39% of cultivated hosts, including bitter gourd, that was the first time reported to be the host of this virus all over the world, and pepper and eggplant and beans, first time reported to be host in Saudi Arabia, while 31% of the samples belonging to different species and different families, these were also identified for the first time as a host worldwide, and we found that TOCV is identified to infect 12 new weed species. CCYV, we found that it is infecting 61% of cucumber samples and 32% of tested weeds. And CYSDV was found to infect 19% of cucumber and 7% of tested weeds. And 9% of these samples had mixed infection with both of these viruses. So CCYV was found to have infection in 4 new weed hosts and CYSDV in 1 new weed species. None of the samples were found to be positive for the infection of TICV and wheat pseudoyellow virus. And the identified whitefly biotype was biotype B, that is now called as MEM1. And 90 minutes of acquisition access period is enough for the whitefly to acquire the virus, and single whitefly is enough to transmit the virus. Again, it needs 90 minutes to inoculate the healthy plants to transmit the virus successfully. And whiteflies can retain the virus for 60 days post-inoculation, post-acquisition, and cultivated hosts are more efficient source of CCYV as compared to the weeds. We have some recommendations on the basis of this finding that a larger number of samples should be collected from all over Saudi Arabia, and if we sequence a full genome, we would be able to find what is the reason that we are having some different results, that TOCV is diverging from other isolates, and we can find what is the strain, and also we can implement the real-time PCR to compare the virus titer in cultivated crops and wheat species to relate the host efficiency of various species and estimate the rate of virus movement in the infected plant. And we can also estimate the efficiency of more weeds as a source of the virus. And as vector is found to be quick in acquiring and transmitting the virus, eradication of weeds would be a better way how to avoid the disease. These are the publications, 2 of the first reports, and 1 paper is already accepted and two papers are under the review. These are the first reports, and we also presented our study in 2 international conferences. One is in Arusha, Tanzania, and the other in Berlin, Germany. So the acknowledgement for I hereby acknowledge my advisor, Dr. Muhammad Al-Saleh, for his very kind guidance and his time, and also my co-advisor, Dr. Ibrahim Muhammad Al-Shahwan, my committee members, Dr. Yunus Yusuf Mulan, Dr. Saud Al-Azmi, and I also thank Dr. Katis, who came from Greece as a foreign examiner for my committee, and Dr. Mahmoud Ahmed Amir, Dr. Jacob Thomas, who helped me in identification at the species level for all the weeds we collected, Dr. Omar Abdullah, Dr. Amjad, Dr. Yasir, Dr. Komi, Dr. Bino Anthony, who helped me with some work with whiteflies, and Dr. Munir Ahmed from Pakistan. He helped me and guided me how to rear the colonies of whiteflies. I also thank my friends Hassan Ahmed, Tahir, Ali, Umar, Dr. Muhammad Altaf, Brother Ammar, Anas, Ahmad Raza, Abdul Rashid Khan, Munawar Manzoor, Khalid, Mureed, Muhammad Naeem, Muhammad Matul Javed, Muhammad Afzal, Hussain Ali, and many others for these 5 years who helped me anyway. Thank you very much.
Okay, now time for a question. Okay.
First of all, I would like to appreciate the work you have done and presented in a nice way, and a very fascinating slide moving around here and there. That is enough to spin a man. So I would like to ask a very simple question from you. What is the reason why you make emphasis on designing the primer on the heat shock region? Number 1. Number 2, in the end you mentioned that when you tried on the datura, I mean the white flies on datura, they are not good to transfer to the next datura plant. So have you ever tried to infect the white flies on the datura and then to transmit them on the other crops?
Okay. Thank you very much for your question. Coming to the first question, we use heat shock protein because when we have to identify the viruses, we go for most conserved region of the genome to identify at species level. we considered heat shock for TOCV, we considered heat shock for CYSDV, and cold protein region for CCY. These all are the conserved regions. And for the second question, the reason why the Datura is not that efficient as compared to cucumber, there are many factors that affects the efficiency of host, like environment, like cultivar, or like the efficiency of the whitefly that is used for transmission. But the most important one is the concentration of virus in the plants. We found from the previous studies that when we implement real-time PCR to compare these ones, we found that the concentration of the virus in cultivated hosts is much more higher as compared to the wheat plants. So we use only Datura to infect Datura and cucumber plants, but other weeds can also be used, and also Datura can be used to infect different cultivated crops also. But the prime thing is the virus concentration in the cultivated host is higher as compared to the non-cultivated crops. Sir.
Just a question about the heat shock protein in the virus. And I would like us to know, for curiosity, what's the function of this heat shock protein for the virus? Second question, the homology of this viral heat shock protein with the other heat shock protein from the other cells. Other like hosts, like for plant heat shock protein, animal.
Yeah.
Homology.
Okay. No, for this there is homology, but up to some extent. I mean, they have to be different anyway from the plants and the, because for plants and other hosts they have other mechanisms also to manage the effect